ஆமாம் இல்லை இப்போ ஆமாம்
Good morning. Good morning. Okay. Hereafter it will not come. Okay. You have given the body. Okay. The other pair in the college is very little. Okay. Good. Sure. Okay, first of all, I would like to thank Dr. Tenmari for inviting me to your talk. And also, I would like to thank the organizers, her colleagues, and also the founder, correspondent, and all administrative people. I thank all of them for giving me this chance to talk something that we are doing at Pondicherry University to all of you. Today I would like to talk to you about the construction of covalent bonds using simple carbene chemistry. Okay, you can use a carbene chemistry to build, to construct, or to synthesize new covalent bonds. Okay, here I am going to talk about few covalent bonds: carbon-carbon, carbon-nitrogen, carbon-oxygen, carbon and sulfur. Okay, so these are the covalent bonds I am going to talk about, and how you can make all those covalent bonds using Catalytic chemistry. This is our research interest. You can see there are so many now. Dihydrogen activation, phosphorylation, carbon dioxide activation and phosphorylation, and then polyethylene degradation, polyethylene degradation, polyethylene degradation. So polyethylene degradation we can do in many ways. One of the ways is to degrade the polymer. You can dehydrogenate. Hydrogenation reaction might have studied. Now what I am saying is dehydrogenation of polyethylene. So you can make double bonds and then you can perform molecular metathesis reaction so that you can break down all the double bonds and make them into polymers. Okay, and then further if you reduce it, you can make a small hydrocarbons which is nothing but fuel. Okay, that's what we are trying to do. And of course, another important research area is excited bond activation. I'm talking about carbon-hydrogen bond activation, carbon-hydrogen bond activation. Okay, so likewise, there are so many covalent bond you can activate and functionalize. And we also work on mechanistic investigation using computational methods. If you want to understand the mechanism of the reaction, you can perform a lot of controlled experiments. In addition to that, if you simulate the mechanistic pathway using computational methods, you can clearly understand what's the possible, really the pathway which the reaction follows. There may be several pathways, but which one is the energetically minimum pathway that one can understand. Another important thing is the organic synthesis. Here I am talking about the new methodologies that we are conducting in our laboratory. And then another important area of research is carbon monoxide releasing molecules. So we can synthesize carbon monoxide releasing molecules. Carbon monoxide, we all know, just carbon and oxygen, right? In organic chemistry, it's simply call it as a ligand. Okay? But the carbon monoxide is a poisonous gas. If anybody takes carbon monoxide, person will die. Okay? It binds with the hemoglobin iron center in an irreversible manner. Okay, once it binds with the iron center, that's all. Oxygen uptake will not be there, so person will die. But if you administrate the fashion with the micromolar concentration of carbon monoxide, it provides a lot of applications. Okay, lot of applications. So we make carbon monoxide releasing molecules and study the ability of releasing carbon monoxide under controlled manner. Okay, that's what we are doing. And then what we do, the carbon monoxide releasing molecule can release the carbon monoxide inside the cell. And you can see what the effect of carbon monoxide inside the cell. I'll tell you one important thing. The heme, if it undergoes decomposition, it produces carbon monoxide. Actually, in our body, carbon monoxide production is always exist. Okay, always exist. This carbon monoxide, when people administrate the cancer patient along with the cisplatin, the normal cell death rate minimized, and only the cancer cell undergoes complex killing only the cancer cell. If you don't provide cisplatin along with the carbon monoxide release molecule, what happens? The person, normal cells as well as the cancer cells will get affected. Okay? So carbon monoxide prevents the death of normal cell, but mechanism nobody knows. So what we want to do is we try to release the carbon monoxide inside the cell and see the effect of carbon monoxide, how it prevents the normal cell death. Okay? When you inject cisplatin along with carbon monoxide releasing molecules. So this is the overview of our research work. Today, what I am going to talk about is only this one, organic synthesis. Within that, I am going to talk about only one particular reaction, okay, where we use the carbene as a precursor to construct new covalent bonds. As I told you, this is the simply shows that we compare technique, competition technique, to carry out the catalysis. Before getting to that, I just want to tell you one important thing. process. Suppose if you want to reduce this cost effect, you have to have an alternate catalyst. But it's we do not have alternate catalyst because we don't know the mechanism to optimize this reaction conditions. Okay, so we need an alternate catalyst. In this regard, if you take or if you synthesize a homogeneous catalyst, it would be easy to understand the mechanism. This reaction is a heterogeneous reaction. Under this high temperature and pressure, we are not able to take out any of the intermediates and analyze and try to understand the mechanism. It's not a problem. Whereas, if you make the homogeneous catalyst, you can thoroughly study the reaction mechanism and you can optimize the reaction and condition so that you can minimize the cost effect. With that reason, several people started working on this, developing a homogeneous catalyst for nitrogen fixation. But everybody faces what is big issue. What is that? When you bring nitrogen and the hydrogen together, in the presence of low oxygen state metal, what happens? The metal is more of basic in nature. It reacts with a relatively more acidic hydrogen molecule rather than reacting with the nitrogen. Okay? So therefore, what happens? When you bring nitrogen and hydrogen, the metal reacts with the hydrogen molecule. Doesn't react with nitrogen. So there is no catalysis. So how to overcome this issue? We came up with a different strategy. Here what we do is, I did not put that molecule. We always take a FLB, frustrated Lewis path. Here frustrated Lewis path. You can take a substituted with large substituted amine and a large group substituted bulky group substituted boranes. So when you bring the borane and the amine together, okay, it forms an adduct. Okay, that is called frustrated Lewis path adduct. When you put the hydrogen gas, this Lewis adduct can split H2 molecule and the proton and a hydride. Okay, now what happens? The reaction which we are having proton and a hydride in the form of salt. There is no free hydrogen molecule exists in the reaction medium. Therefore, there is no combination between nitrogen and hydrogen. Okay, only the H plus and the H minus can react with the water nitrogen. Like this is the mechanism which we have proposed, and we experimentally we isolated the function 5 and we yet to proceed further. Okay, so soon we will surely achieve the entire catalytic cycle. So this is our own strategy. This is the only mechanism available for the nitrogen fixation, particularly when we use of protons and hydrides. Even if we Google it, only our reference will come, and this mechanism is named after our laboratory. Okay, with this, as I promised you, I will get into the promised title, where we are going to construct new covalent bonds. So as I mentioned here, carbon, nitrogen, carbon, oxygen, carbon, sulfur bond, we can construct via the random insertion into XH bond. And the second aspect is the construction of a CC covalent bond via the carbon hydrogen conservation using the Carbene chemistry. Again, you can also convert carbon insertion reaction to CH bond. And thirdly, what I am going to discuss is the carbonization of carbene that can produce a ketene, and the ketene further can react with appropriate nucleophile electrophile to give you the expected product. So these are the things that I am going to discuss. Mainly, when you use a ketene, you can make a beta-lactams. These are all commercially available drugs. Okay, beta-lactams and benzoxazoles. Okay, several benzoxazoles you can synthesize using this strategy. Okay, so that's what I am going to discuss. For better understanding, I will tell you how one can generate a carbene. You can generate a carbene by adding the base to this, appropriate substrate, and then the deprotonates to generate an anion, and then subsequently the Cl minus goes out to generate a dichlorocarbene. Okay, so this is one of the ways to make a carbene. And positive generation and all is like know it from how we can make it, and that is the key thing that we are trying to show. The second important method is to take a dicer compound. Take a dicer compound, you treat it thermally or photochemically. I need a silence. Silence, please. Silence. Early morning I drove car more than 170, 80 kilometers. Okay? So don't make noise. Okay? Yeah. Okay. So you can generate a carbene from dicer compound. Okay, you can take a dicer compound and thermally or photochemically you can treat it, and then you can generate a carbene. And another important ways to make a carbene is take the dicer compound or any other substrate that I mentioned here and add appropriate transition metal complex, or it could be a simple inorganic complex, or it could be a simply organometallic complex, or sometimes it could be simply transition metal salt. Okay, you can make a metal carbene complex like this, and then from here you can generate a carbene. Now you can ask a question, why should I make a metal carbene complex, subsequently release the carbene and use it for subsequent reaction, instead of doing straight away making a carbene like this and using for some transformation? You can ask this question. But the simple answer is, the carbene is a highly reactive species. Remember that, it's a highly reactive species. If it is a highly reactive species, when you treat with some other organic substrate, you won't be having a selective. But you will be getting the products, but you won't be having a selective. That means you cannot have a control over the reaction. So what is the advantage that you are going to have when you make a transition metal carbene complex like this? When you make a metal carbene complex like this, you can have a control over the entire reaction and you can get a better selective. So the best thing is, best way to get selectivity is make a transition metal carbene complex and then release a carbene and then control the manner. Okay? In this way, you can get a high selectivity and you can get a better yield also when compared to the way you make a carbene here and use it for subsequent reaction. So today, as I mentioned, what I'm going to talk about is related to this reaction. I'm going to take a dicer compound and treat with my transition metal salt or complex, and then it can produce a transition metal carbene complex, and then I will add my substrate and see whether the XH bond is undergone the insertion with this carbene to give me this XH product. That's what I'm going to discuss today. Apart from that, the third point which I mentioned in my plan of the presentation is making a ketene. Okay, what I'm going to do here is you can take a dicer compound, treat with the transition metal, and make a transition metal carbene complex, treat with the carbon monoxide, then you get a ketene like this. Once you get a ketene, look at this. The ketene bond is polarized like this. You have a delta minus and delta plus located on this respective carbon. Now, I can add an appropriate nucleophile on this and the electrophile on this to give me the expected product. Look at this. The HOMO is centered on this carbon and LUMO is centered on this carbon. So that means, the nucleophile and electrophile can be added respectively on this and this carbon. And this HOMO-LUMO gap, you see, is roughly close to 4.8 electron volt. Therefore, the ketene is to some extent stable. Okay, you can also deduct this. If you look into the literature, very only few references are available to detect PT experimentally. Okay, we use simply infrared spectroscopy technique to monitor the ketene formation and followed by the product formation. And we prove that our reaction proceeds by ketene formation, none other than that. Okay. Okay, with this I will tell you the transition metal carbenes, how you can make it, and what would be the electron structure of all this transition metal carbene complex. You can see here, this carbene is a sp2 hybridized carbon. Okay, it's a sp2 hybridized carbon. You have one of the sp2 hybridized carbon here, it has 2 electrons, and this one is unhybridized pz orbital. Okay, unhybridized pz orbital. So we call this as a single carbene. If you put the 2 electrons in these 2 orbitals, it's off-filled orbitals. Okay, hybridized orbitals also is there, unhybridized orbital also is there. Now we call this as a triple carbene. And these carbenes can form a complex like this when you add a metal. Look at the electron structure. This is the sigma interaction. This is the Z square orbital which is capable of going for sigma interaction. Can interact with the carbene, one of the hybridized sp3 orbitals. So it can produce a sigma bond. And then this is the metal d orbital which has a pi symmetry. Maybe dxy or it could be xy or xz and yz. And then this one is a pz orbital. So it can go for sidewise overlap to make a double bond. Okay, we call it a pi bond. if you get this electronic structure, we call this as pressure carbene, and often the orbitals we take that's called the shock carbene. Nevertheless, irrespective of whether it's a shock or pressure carbene complex, the reactive pattern is going to be the same, and the stereochemistry probably would differ depending upon the substrate what they are going to use. And then these are the several reactions that Carbene can display. I am going to talk about only the carbene insertion reaction. Apart from that, there are so many other reactions. I am not going to talk about that. And these are the great advantages that you are going to avoid when you make a transition metal carbene complex and subsequently use it for other organic transformation reactions. My reaction condition, excellently, high selectivity, and functional group tolerance. These are the important things you want to perform any kind of organic transformations. Okay? Let us get into the reaction. What I am going to show you is this is the XH bond. It could be a nitrogen or an oxygen. Okay, when you add a diso compound in the presence of a catalyst to make carbene inserted products. And if you look at this catalyst, what would be the appropriate catalyst you can think of? I am going to talk about the copper-based catalyst. We do not have much time to discuss the entire world. To cut short my introduction, I am just mentioning that you can use a copper catalyst. If you take a copper catalyst in the literature, copper-1 basically, carbon, nitrogen, oxygen-based ligands are exclusively used. Phosphorus-containing ligands completely ignored. The simple reason is when you take the phosphine-based ligands to form a complex with copper-1 salt, the resultant copper-1 phosphine complex undergoes decomplexation in solution and it produces free phosphine. The moment it produces a free phosphine like this, it can react with the carbene and gives you air. Okay? So the ligand, it prevents the reaction. It forms a event. Therefore, there will not be a free carbene to react with your substrate. Now, we want to show that how we can make use of phosphine-based ligands. Okay, this is what we did. So this is a special case that we synthesized, and then we treat with the copper-1 perchlorate salt. Reduce you this particular complex, copper-1 complex. Copper-1 in the sense is the oxygen complex plus 1. Okay, not plus 2. Copper-1 complex is always a gas sensitive and moisture sensitive. Nevertheless, this complex is perfectly stable with the moisture and the air. This is the single crystal structure of the complex. You can see there is a copper, okay, it's adopted a tetrahedral geometry, okay, and there is no any other matrix here. So it's clear that we have a copper-1 phosphine complex. Now, if you take this catalyst and take this disocoupled aniline, you get a phosphine inserted product. And this is the substrate scope. I'm not going to discuss in detail, we do not have much time. So you can make a large number of organic compounds and use it for biological studies. And this is the other reaction where we use the oil also. We have a NH2 that's aniline. We know that carbene undergoes insertion in the aniline. If we introduce the alcohol, what will happen? The carbene can undergo insertion in the NH bond as well as OH bond. That's the first question. Second question, can the carbene undergo only NH bond selectively? Third question, or it can undergo carbene insertion only to OH bond apart from that? Okay, this is the third question. Okay, so what we are going to do here is that highly chemoselective manner to do this reaction is more beneficial. So in this regard, what we have done is, this is the reaction optimized condition. You just take a methanol, 2 mole % of our catalyst, this is the one. At room temperature, if you stir it, you will get this product. Okay, the Royce yield, when you introduce the electron donating withdrawing group, you get a Royce yield 0.3%. The electron releasing group that offers you nearly 93% yield. Okay. So electronically, it's very important. The substitution shows clearly more significant in the yield determination. Apart from that, the selectivity. The particular catalyst promotes this reaction in a highly chemoselective manner, it inserts the carbene only in the bond over OH1. Look at the solvent what we use, methanol. Even methanol OH can react with the carbene, but doesn't happen. And also the OH of the substrate doesn't exist. Okay, so aminophenol you can take. Generally, what people do is when you take aminophenol reaction, you want carboxylic reaction, you go for protection and functional protection, reproduction strategy. But now you don't have to apply the functional protection, reproduction strategy. Straight away you can take our chances, add a isocompound, add your aminophenol, and you get one product rather than anything else. So that's the great advantage of this reaction. And then you see, we also use the acid group. Still, when you use the acid group, still our catalyst insert the carbene into only the NH bond, not the acid group. So after benzoic acid, you take only the amine group reacts with the carbene, not the acid group. Okay. Again, this is very important because otherwise you have to go for a protection and reprotection strategy. Okay, that would be more expensive. So you can cut short all those things, straight away take our catalyst and do this reaction. You can get a equivalent product. Okay, so this is another reaction that we have done. And all these products, you can see the similarities in the red molecule what is available in the market. So probably it's all may exhibit excellent biological activity, but all those things need to be studied. And this is the mechanism of the reaction. See, if you have a doubt, you ask me later and give me some time. Don't make noise. Don't make noise. Okay? If you want to ask, if you have any doubt, I will be happy to receive your question and answer. Okay? Don't discuss, don't make noise. Okay? Yeah, this is the catalytic cycle that we propose. You can take the copper-1 complex here, add either alcohol or acid substituted aniline, that means benzoic acid or aminophenol, and then it produces an intermediate 1A like this. You can see here the aniline, the nitrogen is coordinated to the copper-1 and one of the acetylcoordinate accidental goes off, and then this intermediate formation is so important. Okay, computationally we tested this particular intermediate has lower energy than OH as well as COH coordinated one. Okay, if you want to have a OH bond inserted carbene or COH bond inserted carbene product, then the OH should coordinate to the metal. The acid COH should coordinate to the metal. Okay, but these two things are not coordinating because if they coordinate, we should get the other intermediates, but they are not forming. Even computationally, it doesn't exist. When you try to optimize the respective intermediate, the OH coordination with the copper doesn't exist. It goes apart. Okay, that's what it happens. And then what happens? This particular 1A intermediate reacts with the isocompound and produces carbene aniline bound intermediate, and subsequently there can be CN bond formation like this, and then followed by proton migration that gives you the expected chemoselective product. Now the question is, why this 1A is possible? What is the importance of copper-1 here? Why the copper-1 does this? The simple answer is, you might have studied, let's say, the principle. Okay, hard and the soft, acid-base principle, right? Okay, Pearson's principle. What it says, the copper-1 is a soft chemical hardness of the copper-1 is highly soft. That's what all the textbooks write. But in my personal or in our group where I did my PhD, that time also we realized this concept. That means the copper-1 is not just that soft as the nickel-0. Nickel-0 is a very soft in nature. It's not that soft. Again, that means is it odd? No. The copper-1 again is not odd as zinc 2+. Okay, zinc 2+ is very odd. It is not that hard. Again, it is not that soft as nickel-0 does. Okay, that means it falls in the borderline case. It falls in the borderline case. So what? So what? If it is fully odd in nature, it will prefer all odd ligands. If it is a fully soft in nature, it will prefer all soft ligands to be around. Since it is a borderline case, it prefers two soft ligands. So many experiments say that there should be a minimum one odd ligand to separate copper-1 intermediate. And then what is the nature of that odd ligand? It should be odd as oxygen? No. It could be a soft or odd. Okay, I'm not confusing, but if you don't get it, ask me later. Okay, so that means it's not odd as oxygen. Therefore, the copper one quickly cleverly prefers nitrogen to be in the coordination sphere, not the oxygen. Okay, because this coordination provides more stability to the system. There is one more rule called principle of maximum hardness. Principle of maximum hardness. Any chemical entity should reach the maximum hardness in order to be more stable. So this strategy helps here, this particular intermediate, to have a better stability, and therefore further can react with the dicer compound to be the carbon complex, and further can be the electromagnetic reaction with the amine, the FH bond, and then as I mentioned, the proton migration produces the expected product. So this is the mechanism that we proposed, and we published this in the Journal of Organic Chemistry. It's a JCS journal. We use all this work. And then what we have done here is that we introduced the electron withdrawing substitute in the dicer compound. There are in the major class of dicer compounds, if you see, there are three. One is electron donating group substituted Rb. So both the side here and here, electron donating group can be there. Another one is one side electron donating, another side electron withdrawing. Third one is this one, electron withdrawing and electron withdrawing. What the chemists they practice? Chemists will practice only electron donating and electron withdrawing, because this is not very stable, at the same time not too reactive. It's a moderate. Therefore, you can easily handle such a substance. What will happen if you take electron donating and electron donating substituted carbene? It's a highly reactive. You cannot even synthesize. The moment you synthesize, decompose and produce a carbene, immediately converts into self-dimerization. We call it as and produces the olein. Okay, you cannot do any other reaction. But if you take this type of substrate, what is the disadvantage? Nobody touches this type of dicer compound. The simple reason is it's highly stable. It doesn't even produce a carbene. Okay, and we have taken the challenge. Let us see whether it can produce a carbene under our conditions. So what we have done is we have taken the iridium not complex as a catalyst and carried out this reaction, okay, in water mainly. Okay, take the water, put this complex, put this dicer compound and aniline. You can see carbene insertion reacting nicely. Takes place, gives you a large number of products. Okay. Again, steric factor, electron factor, seriously affects the limit of the reaction. I'm not going to talk about that because we might have such time. And we published this work in Organic Biomolecular Chemistry. It's RS journal. And then we also introduced the phosphonate group in the dicer compound. What is the great advantage of using the phosphonate? The phosphonate containing organic molecule shows a large amount of biological activities. In fact, protein labeling and all is important. So phosphonate people want to add to the protein so that they can monitor the protein folding and etc. It also offers a kind of fluorescence property. All the things can be done. Then you add appropriate transition material to this phosphonate. Okay, so there are so many steps to do. But labeling the protein with the phosphonate becomes so important. And also the important thing is the protein is comfortable in water medium rather than either solvent. So this strategy gives you way to even label the protein in water, because this reaction works in water. You can take a solvent as water and perform this reaction. Look at the catalyst. Simply, copper 1 astronaut. That's all. We don't have to add anything else. Okay, nicely that produces a carbene inserted product. You can see a large number of the substrate scope we have here also. We can tune the aniline also here. Here the phosphonate you can tune, and here aniline you can tune and get a different organic product. And these are all really very useful for biological studies, and we are doing that now. And apart from that, this is the other type of substrate that we use. You can see the similar backbone is available already, known that in the market. Okay. So this aspect, what are the compounds that we produce is a very important one. And this is we can use our strategy to make substituted immunosomes. We can make several substituted immunosomes using this particular strategy. Again, we can take the dicer compound. You can treat with hydroxide, and that gives you cyclic, heterocyclic product like this. So again, the mechanisms are so interesting. If you don't have any time to discuss, I will pass this. Okay, so this done by Bilal, and we published this in the Journal. Apart from that, as I mentioned, the diso compounds are not that stable. Okay, are not that stable. Whenever you need a carbene, you have to make this diso compound use it. None of the diso compounds are commercially available. Note this down. None of the diso compounds are commercially available. Okay, again, you cannot make it in a large quantity. Now, what we have done here is that you can take the diso compound, simply add copper powder in the DMSO, okay, at the 50 degrees Celsius, you get sulfoxylic gelate. You might have studied the Fourier reagent. It's one of the Fourier reagents is called methylene sulfoxylic gelate. Okay, it's a carbene source, and synthesis of methylene sulfoxylic gelate, if you look at the procedure, it offers only 33% yield, not more than that. Photochemically and thermally, you have to do this reaction that offer you only 35% yield, not more than that. Look at our reaction. You can get the excellent yield. Okay? Again, the condition is very simple. Just take a copper powder and put it in the reaction, and then DMSO as a solvent, just stir it. Okay, at 50 degrees Celsius, so you get a sulfoxylic gelate. So what? You got sulfoxylic gelate. What is the great advantage now? I'll tell you. You can make it in kilos. Sulfoxylic gelate you can make it in kilograms and can store it in the bottle, keep it in the shelf. Whereas diso compound, none of them are stable. Always, whenever even if you make it, you have to put it in the refrigerator. But now you can store the sulfoxylic gelate in the bottle, keep it in the shelf, keep it in the some salt in the laboratory. Okay, and when we demonstrated that, we can also use it as a quantum precursor, and at this particular reaction condition, something degrees Celsius is only when you use a scanning as a catalyst, you can produce a carbene, and then carbene can insert with the NH1 as we demonstrated earlier, and it can give you the expected product. So what I am trying to say here, the sulfoxylic gelate can serve as a carbene precursor as diso compound does. Okay, so that is my conviction, and that's what I am trying to say here. And this is the simple mechanism. Already I discussed about this, but here the scandium can be used as a catalyst. And the mechanism you see carefully, the carbene complex of scandium formation is so important, and further it can interact with the nitrogen and it forms a CH bond formation, and then further proton migration takes place to give you the expected product. And then what we have done is, can it be used to insert the carbene to OH bond of acid? Yes, you can do that. And you can get a carbene inserted to OH bond of acid that gives you this particular product. So we say a large number of products here. Again, we published this paper in Journal of Organic Chemistry, and it's appeared on the cover page of the journal, and Carbene worked on this. And again, you can also make a CS bond, and this is the new CS bond that we synthesized. So you can take respective thiol and add a diso compounds, and then you can make respective this product. Okay. So this type of compounds are really very useful. You can see the backbone of all these drug molecules, particularly this oxidose or there. Okay, so this is what is so important. Okay, you can make ethers and synthesize biologically relevant molecules in a simple way. Okay, and then this is the substrate scope that we have done. And then this is the mechanism of the reaction, and these are the important molecules already known. That's also okay. With this, I just want to show you another important reaction. You might have studied the Beck reaction. Beck reaction is nothing but molecular arylation. Okay, it's got a Nobel Prize. Okay, just add a palladium, take aryl halide, and add a styrene that will offer molecular arylated product. It's got a Nobel Prize. Okay. Now, if you want to alkylate it, what is the solution? There is no solution. How will you alkylate the molecule? Okay. It can tell you only arylate. How to alkylate? I'll tell you how to do that. You take this diso compound. It selectively inserts carbene into the CH bond of the terminal olefin that gives you this product. Okay. So remember that this is the kind of alkyl group that you can introduce into the styrene, particularly in the terminal olefin carbon. Okay. We also can call it as a CH bond functionalization of terminal olefin. Okay, these are the important strategy. Okay, for synthesizing carbonic backbones, where a lot of biological applications we can derive from that. And this work, we published in JOC. Dr. Deepak Group, they joined with us at the time, and one of our students also joined and did this work in this related area, and they are there in the author list also. And then this is the mechanism that we proposed. It's a very interesting mechanism, but we do not have time. Sorry. Okay, very interesting mechanism because I did not tell you one thing. Can you see here? There are two things are there. Okay, this is a plus, not this alone. Okay, this is a plus. But how to make the stereo? What is that called? Chemoselective reaction. How to do that? And sometimes it gives you a single product. Sometimes it gives you AB mixture. Okay, and how to do that? What is the key intermediate that forms to give you a single product? What is the key intermediate that forms to give you two products? What is the key intermediate that forms for formation of 1A or 1B? The number is given here, don't worry about it. Okay? So this is the very important thing, but such an interesting mechanism is there, but I don't have time. Okay? I'll just pass it. So this is another important reaction. So you've done. You can take a NN bond. You can easily make NN bond, and this is a very interesting mechanism that we propose. In fact, this type of intermediate what we propose, nobody convenes. Okay, I have taken it from my supervisor, Professor Ashok Samhansan, IIT Bangalore, to support me in this regard. Okay, I went to Bangalore to discuss about this mechanism, particularly for this intermediate. Okay, I visited him to discuss about this mechanism, particularly to propose this particular intermediate. Okay, it's not that easy to form this intermediate. Reason is, number one, always the moment you put a diazo compound, it forms a carbene. But here there is no carbene formation. How the copper one forgets its function? How it forgets its function? Who made copper to forget its function? There is a way to do that. This is the key thing that reacts with this copper one reacts with the magnesium salt that is available here. Okay, so this intermediate formation is very important. Here the diazo compound directly interacts with magnesium rather than the copper, and that gives you the expected NN formation. It's called a diazine. It has a lot of photochemical and physical properties. Okay, so that's what it is. Anyway, I don't have time, and again. She worked on, Gregory worked on this. Slim substituted the olefins, and he also synthesized a similar product as I told you related to Carbene. Okay, here also we made a large number of substrates, and this is again very interesting mechanism. Here the key intermediate we propose here, the allyl type of intermediate formation is quite interesting. Okay, so how the anion shifts whether to this carbon or this carbon, that will lead to the formation of chemoselective products. If you don't have time to get into the details, this also will publish on the cover page of the article. Okay, and then the ketone formation, I don't have time to stop. The ketone formation we have done thoroughly here, and I will just show you how the ketone can be useful. Amido phosphonate we can make from this reaction. I don't have time to say all the things. Then again, the ketone formation is useful to make beta lactose, as I promised you earlier. Is the way to make beta lactose. It's also published on JOC. And this is the simple mechanism that we propose. This also appeared on the JOC cover page article. Okay, and this is another important reaction where we can make this carbon substituted product. Okay, so this again benzazole are very important. And you can make it in the two steps. Okay, first step, just take a diazo compound, get this intermediate, then treat with the T, and then under the Celsius, you can get the cyclic product. Okay, this is the reaction. This molecule, you can see there is a drug molecule available in the market, and also has a similar structural features. Therefore, we can also expect better good biological activities for these compounds, and yet to do all this. So we publish this in more synthesis and catalysis. This is a widely synthetic journal, it's an important journal again. And this is the mechanism of this reaction. Okay, we don't have time. Okay, and these are the compounds that we synthesize with the main. Each and everything is a new strategy to make all these type of molecules. I've shown you a small portion of what we have done, particularly carbene related reaction to one synthesis reaction. Okay, with this I would like to thank all the funding agencies, our university, and this is the CSIR, and this one is the ANRF, Commission on Research National Foundation, and then the SARB and UGC for the generous funding. We got a lot of funding actually. Some projects we have completed. Okay, and these are all our contract students. Okay, from our laboratory, they are working in the different companies or they are all faculty in some other institutes. With this, I would like to thank you for your kind attention. This is our we have a big labs and we have so many equipments. Okay, everything is there in our laboratory and in our department and also in our university. Whatever the names, we do have it. Okay, so the excellent the infrastructure we do have on the table. So with this, so I thank the organizers for giving me this chance. Thank you.
Thank you for bringing your presentation.